The Calvin cycle is the second main stage during the process of photosynthesis. And it was known a long time ago, back when I was a student, as the dark reactions because they didn't require light, but the name was kind of confusing. And sometimes students thought that the light reactions happen during the day, the dark reactions at nighttime, and that is not the case. Both the light reactions and the Calvin cycle will be happening during the daytime for most plants on planet Earth. And so, what powers the Calvin cycle? Well, we know that the energy is in the form of ATP and it comes from the light reactions. And we need also high energy electrons transported by NADPH, and those also come from the light reactions. So at the same time, the light reactions are producing ATP and loading electrons into NADPH. That's when the Calvin cycle has to be happening. And again, we have some exceptions, but for most plants, this is going to be the case. And the ultimate goal of the Calvin cycle is going to be making a sugar molecule. First, we have a molecule of G3P, glyceraldehyde 3-phosphate. That's what comes out of the Calvin cycle. And eventually, uh, two molecules of G3P will combine to make one molecule of glucose, which is what we promise in our overall equation for photosynthesis. So let's break down the Calvin cycle into steps. The first step is going to be carbon fixation. This is going to be when a specialized enzyme, the enzyme is called Rubisco. Rubisco is kind of like a short nickname for ribulose biphosphate carboxylase. Because that name is too long, we just conveniently say the name Rubisco, but I do want you to know that Rubisco stands for ribulose biphosphate carboxylase. And so the reason it has this particular name is because the enzyme Rubisco is going to take the 5-carbon sugar ribulose biphosphate and it's going to fix. Fixing here means like sticking, like attaching, like when you fix something to a wall, means that you're attaching it there permanently so it doesn't fall off. So carbon fixation means attaching carbon from carbon dioxide onto this 5-carbon sugar called ribulose biphosphate. And the first step is going to be producing a 3-carbon sugar known as phosphoglycerate. And this phosphoglycerate is a very unstable compound, and it's going to be quickly changed by ATP into electrons from NADPH into a G3P molecule. So before we move on from this first step of carbon fixation, I want to mention to you that Rubisco has some interesting details about it. It has limitations, and one of those is going to be that Rubisco can only incorporate 1 carbon from carbon dioxide at a time. So every time the Calvin cycle goes around, Rubisco can only bring 1 carbon molecule. So in this illustration, we're showing an input of 3 CO2s, meaning that this cycle has to go around 3 times before those 3 carbon dioxides can be incorporated. Sounds confusing? Well, I hope it isn't. You just imagine like Rubisco was this one-handed molecule. Imagine like the CO2 like a ball, and you can only have one of those balls at a time, moving them, giving it to ribulose biphosphate. Another interesting detail about Rubisco is that it's kind of like an imperfect type of a protein, because even though it helps with carbon fixation, if oxygen is present in high levels, it will fix oxygen instead of carbon dioxide. And so that's a detail that I will be bringing back to your mind in the next video when I talk about carbon fixation strategies in plants. So just for right now, remember that Rubisco is kind of like an imperfect molecule. If carbon dioxide is available, it fixes carbon dioxide. If oxygen is in abundance, then it does that oxygen fixation, which is not at all what we want, not at all. One more detail about Rubisco, which I think is interesting. Your textbook mentions that Rubisco is believed by biologists to be the most abundant protein on Earth. And they just say that. They don't give any more explanations. And so I think the rationale for saying that Rubisco is the most abundant protein on Earth is that because so much of life depends on chemical energy, which was before becoming chemical energy, light energy coming from the sun. And because so much life depends on those glucose molecules that are made in the Calvin cycle, that scientists go on to say that Rubisco is the most abundant protein on Earth. When you think about it, the glucose molecules, which were the foundation for other molecules in the food we eat, had to go, the carbons in those glucose molecules had to go through this enzyme called Rubisco. And if it's so much to think about, if your mind is going like right now, like, well, don't worry too much about it. Just an interesting thought, in my opinion. But anyway, you can spend some more time thinking about it. The next step in the Calvin cycle is going to be reduction. Reduction means that electrons carried by NADPH will be given to these intermediary compounds we have here in the Calvin cycle, and eventually a G3P molecule will be made. Now, remember the detail I told you about Rubisco being able to bring in only one carbon at a time into the Calvin cycle. In order for us to take out this one molecule of G3P, the Calvin cycle must run around 3 times. Okay, let's think about this. What would happen if you bring only one carbon, but you're taking 3 carbons out? Do you need an analogy? Okay, so imagine that you have a bank account and you open the bank account with $1, but then hours later you want to go and withdraw $3, but you only put $1 to begin with. Will the bank allow you to withdraw $3? And then you want to do it again. You go and deposit another $1, but you want to take out 3. You can't do that. You're going to be in debt. And the same thing happens with carbons in this cycle if you only bring 1 carbon at a time, but you want to take 3 at the other end. And so to avoid any confusion, you can't do that. In order to take 3 carbons out in the form of a G3P glyceraldehyde 3-phosphate molecule, there has to be 3 runs of the Calvin cycle, each time bringing 1 CO2 molecule. So when the Calvin cycle has run around 3 times, then we take out 1 G3P molecule. And the last step that we need to have here is regeneration, the remaking of ribulose biphosphate. And this has to be rebuilt because it is needed at the beginning of the Calvin cycle for Rubisco to continue fixing 1 carbon into ribulose biphosphate. So there you have it. That's the Calvin cycle. And one of the main lessons to remember is that Rubisco, which fixes carbon, is going to allow for only 1 carbon to come at a time. So the Calvin cycle has to go around 3 times in order to make a molecule of G3P. Eventually, this can happen in the chloroplast. It can also happen in the cytoplasm of a plant. 2 G3P molecules will be combined in the making of 1 glucose molecule. The math is simple. If 1 G3P molecule has 3 carbons, and we combine it with another G3P molecule with 3 carbons, 3 plus 3, that's 6, and that's what you need in a glucose molecule. The formula for glucose is C6H12O6. So to have 6 carbons, you need to join 2 of these molecules of G3P. And that's the Calvin cycle. I will make one more video to explain to you more of the nuances, more of the details involved with the carbon fixation process and how different plants can accomplish this important step of the Calvin cycle.